So I just want to welcome everybody to our 811th meeting of the Eastern PA branch of ASM. This is our annual student chapter organized May monthly meeting. We apologize for the last minute change to virtual, but luckily we're all kind of used to Zoom by now. So this should still be a great meeting, and we're very happy to share our excellent set of speakers with you today. To start with, we're going to hear from three graduate students. These three students won our top poster awards at our student chapter organized Philadelphia Infection and Immunity Forum in December. These students demonstrated excellent research and presentation skills. So every year we like to give them a chance to share their work with the entire branch in this format. So for our talks today, please feel free to ask questions in the Zoom chat and we will relay those to our speakers, or you can raise your hands or unmute yourself after the talk is done and unmute and ask your question. So without further ado, I will turn it over to Zach to introduce our first student speaker.
Thanks, Caitlin. So it's my pleasure to introduce our first speaker, Daniel Netting. Dan is a third-year PhD candidate in the Immunology and Microbial Pathogenesis program at Thomas Jefferson University. He's in the lab of Dr. Adriana Montagaza, and he's going to share his talk with us today titled "The Phagolysosomal Nucleoside Transporter SLAC29A3 Regulates Inflammatory Responses in Dendritic Cells." Take it away, Dan.
All right, thank you very much, and thank you for having me. I am really grateful to be able to be here and give this talk today. Let's see. Okay, can you see my screen and hear me all right?
Looks good to me.
Awesome. All right, well, hi, everybody. Like Zach said, I'm Dan Netting. I'm a third-year student at Thomas Jefferson University in Adriana Montagaza's lab, where we study the role of solute carriers in dendritic cells. So I'll be sharing my research on the solute carrier SLAC29A3 and its regulation of inflammatory responses in dendritic cells. I'm going to change this to a laser pointer. There we go. Okay, so inborn errors of metabolism are diseases which affect the body's ability to metabolize and utilize nutrients. Individual IEM diseases are relatively rare, but the umbrella of IEM diseases is fairly common, occurring in about 1 in every 2,500 births. Some of these diseases are caused by mutations in a family of proteins called solute carrier transporter proteins. Solute carriers, or SLACs for short, it's a superfamily of over 400 different transporter proteins. These are membrane-bound transporters, which, as the name suggests, transport solutes across a membrane. Over 50 of these are currently associated with disease, and that number continues to rise as time goes on. These solute carrier transporters can localize to a number of different organelles in the cell. A lot of them are seen on the plasma membrane. You'll also find them on mitochondrial membranes and lysosomal membranes, as well as other organelles. So this here is a non-exhaustive list of solute carrier proteins on the lysosome, which are associated with disease. So you can see there's a lot of these, and these are just lysosomal ones associated with disease. And the list actually even goes further than that. We are really interested in this one right here, SLAC29A3. This is a nucleoside transporter found on the lysosome, and it's actually the only intracellular nucleoside transporter that has been described. The SLAC29A3 preferentially transports nucleosides in the cell, but it is also capable of transporting other solutes. It's been reported to transport nucleotides and bile acids, for example. The dysfunction or mutations in this protein causes a human inflammatory disease called H syndrome. H syndrome is characterized by symptoms such as hearing loss, hepatosplenomegaly, histiocytosis, hypertrichosis, and really a lot of symptoms that begin with the letter H, which is where it got its name from. Pictured here is an H syndrome patient who has severe arthritis, causing their fingers to curl or their joints to curl. And down here is an H syndrome patient with the characteristic hyperpigmentation in his legs, as well as patches of hypertrichosis. Interestingly, there are a number of diseases that were previously not associated with SLAC29A3 or H syndrome, such as Rosai-Dorfman disease and Physalba histiocytosis. These are both lymphoproliferative diseases. But as time went on and there was more genetic evidence of these diseases, they are now linked to SLAC29A3, suggesting that this is a more common disease than people realize, and that it's kind of this umbrella disorder of SLAC29A3 related diseases with varying degrees of severity. Our lab first became interested in this gene during a screen of the dendritic cell phagosomes for proteins on the phagosome involved in inflammasome activity and in pro-inflammatory signaling. So SLAC29A3 was one of those hits in the screen. And indeed, when we examined these dendritic cells and transduced them with a SLAC29A3 GFP expressing construct, and then pulsed those dendritic cells with Texas red beads, you can see that protein SLAC29A3 is nicely localizing to the membrane of the phagosome and is even on phagosomal tubules. So we also have proteomic data to show that it is on the phagosome. So it's clearly there on the phagosome, and we've started to research it. Importantly, SLAC29A3 has been investigated in a number of other immune cells, including T cells, macrophages, and monocytes, but it's never been investigated in dendritic cells before. So this slide is kind of an overview of my project and what I'll be discussing. I know it's a bit busy, so I'm going to go through it a piece at a time, starting with the pH story. So when a dendritic cell or a phagocyte encounters a particle such as a bacterium, it will engulf it in a process termed phagocytosis, leading to the enclosure of that particle in a de novo organelle termed the phagosome. The phagosome can then mature through the fusion with endolysosomal organelles, such as endosomes and lysosomes, and it acquires the content of these organelles in that process, including endosomal pattern recognition receptors like TLRs, lysosomal degradative enzymes, and lysosomal solute carriers. Within the phagosome, that particle, such as the bacterium, will be progressively broken down by those lysosomal degradative enzymes, generating things such as antigens for the presentation to antigen-specific T cells, as well as PAMPs for recognition by pattern recognition receptors and subsequent induction of immune responses, as well as a number of different solutes, such as nucleosides, amino acids, and ions. These solutes that are being generated need to be exported out of the phagolysosome, or else they'll begin to accumulate. And their accumulation is predicted to have a number of consequences within the phagosome. For one, several of these solutes, in this case, I'm focused on SLAC29A3, so I'm going to be focused on nucleosides. Nucleosides are going to, or are predicted to affect the pH of the phagosome, as they are slightly basic and will interact with free protons, thereby alkalinizing the phagosome. So we sought to address this by assessing phagosomal pH in splenic DCs. We isolated the spleens of wild-type and SLAC29A3 knockout mice, and then we pulsed those splenic DCs with beads that are coated with a pH-dependent fluorophore FIT-C and a pH-independent fluorophore Alexaflor 647. And we can then read the fluorescence out by flow cytometry. So pictured here is just a picture of what the kind of flow plot looks like. So you can see there are these individual populations depending on how many beads the dendritic cells have taken up. So we can gate on one of those populations, and we can then look at the MFI of both the pH-dependent channel, FIT-C, and the pH-independent channel, APC, which is Alexaflor 647. And we get this ratiometric readout that we can compare to a standard curve to get an idea of what the phagosomal pH is. We can get a number with that standard curve. And as expected, through this assay, we found that the SLAC29A3 knockout splenic DCs have an increased phagosomal pH. They're more alkaline than their wild-type counterparts, which holds true even as the phagosome progressively acidifies as it matures. This change in pH is really important because a lot of these degradative enzymes within the lysosome have an acidic pH optimal. They function best at acidic pHs. And so we expect that this alteration in phagosomal pH will impair the activity of these enzymes. For one, the degradation of the cargo, such as the bacteria, into its component parts, including into PAMPs, is done by these degradative enzymes. And so if these enzymes are less functional due to changes in pH, then we expect there to be fewer PAMPs generated. Additionally, some pattern recognition receptors, such as endosomal TLRs, require cleavage by these lysosomal degradative enzymes for their proper conformation. We expect pH, this alkalinization of the lysosome, to impact the generation of TAMPs as well as the activity of these TLRs. And indeed, when we stimulate wild-type and SLAC29A3 knockout bone marrow-derived dendritic cells with Salmonella typhimurium, either attenuated live Salmonella or gentamicin-killed Salmonella, we see that there is this pretty significant reduction in cytokine secretion, both IL-6 and IL-12, in response to the bacteria. We confirmed that this was a defect due to SLAC29A3 through a rescue experiment. So we transduced wild-type and SLAC29A3 knockout BMDCs with an empty vector and assessed the cytokine secretion. And as expected, there's a significant drop. And then we also transduced the wild-type and SLAC29A3 knockout BMDCs with a SLAC29A3 GFP construct to rescue expression of this protein. And you can see that the rescue increases the cytokine secretion of the SLAC29A3 knockout cells to about the same level as wild type. Interestingly, the wild type BMDCs, when they have this overexpression of SLAC29A3, do also get this increase in cytokine secretion, but the fold change between the empty vector and the 29A3 GFP and the wild type is less than the fold change in the knockout, still suggesting that there is a rescue. We further interrogated this through some nuclear fractionation experiments. We stimulated wild type and SLAC29A3 knockout BMDCs with a higher MOI bacteria. This was an MOI of 30. And we performed nuclear fractionation to assess the proteins in the cytosolic fraction and in the nuclear fractions. And we found that NF-kappa B is, as expected, translocated to the P65 subunit of NF-kappa B is translocated to the nucleus in response to bacterial stimulation and wild-type BMDCs. But we see there's less P65 translocation occurring in the SLAC29A3 knockout BMDCs, suggesting that SLAC29A3 does in fact promote NF-kappa B activation. Now, of course, there are other inflammatory pathways that could be at play here. So we also assessed the AP1 pathway, but we found that the stimulation of wild type and SLAC29A3 knockout BMDCs with Salmonella did not cause any changes in the phosphorylation of the MAP kinase P38, which is right upstream of AP1. And when we did that same nuclear fractionation experiment, June, which is one of the components of AP1, is translocated to the nucleus to the same degree in the wild type and the SLAC29A3 knockout cells, suggesting that SLAC29A3 doesn't promote the AP1 pathway, but does promote the NF-kappa B pathway. The next thing we were looking at is this transcription factor TFEB, and I kind of want to run through the activation of TFEB really quick because it is not a super commonly described transcription factor. TFEB is known to be the master regulator of autophagy and lysosome biogenesis. And normally, TFEB is phosphorylated in the cytosol, and its phosphorylation enables interaction with these 14-3-3 proteins that keep it sequestered in the cytosol. However, phagocytic events cause the activation of this calcium channel mucolipin-1, and the activation of that calcium channel allows for calcium efflux, which activates the protein, the phosphatase calcineurin. Calcineurin has many targets, one of which is TFEB. So this calcium flux activates calcineurin, dephosphorylates TFEB, and allows TFEB to shuttle to the nucleus where it activates genes related to autophagy, lysosome biogenesis. And more recently, it's also been described that TFEB regulates the production of pro-inflammatory cytokines. Now, we're really interested in TFEB because it's been found that that calcium channel, mucolipin-1, that is required for TFEB activation is inhibited by the presence of lysosomal adenosine. And so we think that in SLAC29A3 knockout DCs, that adenosine that is being accumulated within the phagosome is then able to go and interact with mucolipin-1, but that calcium channel, and block its activity, thereby blocking that calcium efflux and the subsequent activation of TFEB and the downstream effects of cytokine secretion and lysosome biogenesis. To assess this, we transduced wild-type and SLAC29A3 knockout BMDCs with a TFEB GFP construct, and then stimulated it with live attenuated bacteria for 3 hours, and looked at the translocation of TFEB to the nucleus by fluorescent microscopy. You can see in this image that in the wild-type cells, there's a lot of TFEB in the nucleus. You can see these bright dots. This is TFEB being translocated to the nucleus compared to the more dim cytosol. Whereas in the SLAC29A3 knockout cells, the majority of the cells have TFEB in the cytosol without that translocation to the nucleus. We've quantified that here, so you can see there is significantly more cells that have nuclear TFEB with the wild-type BMDCs compared to the SLAC29A3 knockout BMDCs. We further confirmed this with nuclear translocation experiments, where we once again stimulated wild-type and SLAC29A3 BMDCs with attenuated salmonella type murium, and we assessed the translocation of TFEB to the nucleus. And you can see that TFEB in the cytosol, in the presence of bacteria, there is less TFEB in the cytosol of the wild-type BMDCs as that TFEB is getting translocated to the nucleus. On the flip side, in the 29A3 knockout BMDCs, you don't really see that translocation, suggesting that there is less TFEB activation, and that SLAC29A3 is promoting TFEB activation to allow it to shuttle to the nucleus. So some of our data points to a reduction in pro-inflammatory cytokine secretion, although we do also believe that there may be a reduction in anti-inflammatory cytokine secretion. But nevertheless, the patients who have SLAC29A3 related disorders present with hyperinflammation. And we desire to interrogate where that hyperinflammation is coming from. And considering that TFEB is the master regulator of autophagy, we became interested in this. Autophagy is the process by which the cell is able to clear out material such as damaged mitochondria, but it is known to be an anti-inflammatory process. And so you can imagine that a defect in autophagy would cause more inflammation within the body. So we see, as expected, when stimulated with bacteria in wild-type BMDCs, TFEB is rapidly dephosphorylated, whereas in the SLAC29A3 knockout BMDCs, there is less dephosphorylation of this TFEB. We expect this to impact the induction of autophagy. We assess this by looking at this protein LC3. LC3 is a protein which is found in this unlipidated form in this top band right here. This is LC3-1. This is found in the cytosol, but upon the induction of autophagy, LC3 gets lipidated and is found on the membrane of autophagosomes and forms this lower band down here. And so an increase in this lower band down here suggests the induction of autophagy. And you can get a ratiometric readout of this lower band LC32 against the upper band LC31 to get a measurement of autophagy induction. And when we do this, we see that SLAC29A3 knockout BMDCs have a significantly lower ratio compared to the wild-type counterparts, suggesting that there's less autophagy induction going on. We also assessed autophagic flux, or the progression of autophagy, through this protein P62. As autophagy progresses, P62 is progressively degraded. And so you can see in the wild-type BMDCs, after the addition of bacteria, which induces autophagy, P62, the amount of P62 is going down, suggesting this autophagic flux is occurring. Over in the SLAC29A3 knockout BMDCs, we actually begin to see an accumulation of P62, suggesting that there is less autophagy occurring than in the wild-type BMDCs, again suggesting that there is a defect in autophagy in these cells. So in conclusion, we found that SLAC29A3 promotes the maintenance of an acidic pH in the dendritic cell phagosome. It promotes the secretion of pro-inflammatory cytokines in DCs. We also have evidence that it promotes the secretion of anti-inflammatory cytokines, but that's another story. We found that it mediates, or it promotes the activation of TFEB in response to phagocytic events, and that SLAC29A3, through this promotion of TFEB activation, also promotes the induction of autophagy. I just want to thank my lab members, the Jefferson Core facilities that assisted us with these processes, our collaborators, Dr. Portolano, Dr. Roche, Dr. Eisenlohr, my thesis committee, and Dr. Brodsky for providing us with those salmonella strains that we use for these experiments. And I'm happy to take any questions. Thank you.
All right, great talk. Thanks, Dan. If you have any questions, you can go ahead and drop them in the chat now. I'll get started. I think we have time for one or two, maybe. Dan, do you have any, I guess, intentions of looking at like peptide presentation from antigens that might be processed worse in the SLEC 29A3 DCs?
Yeah, so we are going to shift our focus towards antigen presentation pretty soon. So we've started to do some experiments like the presentation of the model antigen E alpha. So we're looking at that. But to your point, one experiment I'm interested in doing is to assess the activity of like lysosomal cathepsins to see if there's any change in their activity and if that is affecting antigen processing, because that would then, in turn affect antigen presentation.
Got it. All right. If there's no further questions, then I think we can turn over to our next student speaker. Thanks, Dan.
All right. Hello, everyone. For our next and final speaker of the day, it's my pleasure to introduce one of our very own student officers, actually, Jadine Sewell. She is a PhD candidate in Sunny Shin's lab, and she's going to be talking about some of her research today with her talk titled The Coxiella burnetii effector that interacts with the host PAF1 complex and suppresses innate immune responses.
Thank you, Adam. Let me share. Can everyone see my slide okay? My slides?
Yep, looks good.
Perfect. Hi, everyone. Thank you for that introduction, Adam. Today, I would like to talk to you guys about some of the work I've had the pleasure to work on in Dr. Sunny Shin's lab. As Adam said, I'm a third-year graduate student here at UPenn. And today, I want to highlight to you this Coxiella effector that interacts with the host PAF1 complex and suppresses the host innate immune response. Before I get into that, I wanted to give you a little bit of background about the pathogen I work with. Coxiella is a gram-negative bacterial pathogen that causes the disease that typically infects livestock animals such as goat, cows, and sheep. Humans typically get infected with Coxiella through the inhalation of contaminated dust or hay particles. Upon inhalation, Coxiella causes this flu-like illness known as Q fever. Roughly about 60% of Q fever cases are asymptomatic. However, 40% of these Q fever cases could lead to infection, while 2% lead to chronic infection that could last up to 5 years. Upon inhalation by humans, Coxiella typically infects alveolar macrophages in the lung, where it replicates in this large phagolysosomal vacuole known as the Coxiella-containing vacuole, CCV for short. Coxiella is a stealth pathogen as it has a high environmental stability where it could travel up to 200 kilometers on dust particle. It has this low infectious doses where one bacterium is enough to cause infection. Due to its large tail transmission and its potential to be used as a bioweapon, Coxiella is labeled as a Category B select agent. Taken together, Coxiella is a poorly understood pathogen, and due to the current treatment regimen for Q fever that requires 2 to 3 years of antibiotic treatment, there's a better need to understand Coxiella pathogenesis and a need to find better therapeutics. Now, upon invading the macrophages, Coxiella utilizes this type 4 secretion system to secrete over 100 protein effectors into the host cell cytosol. These effectors are important for the bug to set up its replicative niche and evade the host immune response. This immune evasion is important for Coxiella as it could take up to 4 days to replicate in the cell and set up its replicative niche. The mechanism of some of these effectors have been shown to modulate different immune pathways such as apoptosis and NF-kappa B pathway in humans, as well as caspase-11 activation, inflammasome activation in mice, as well as MAPK pathways in yeast. Now, taken together, Coxiella activates multiple receptors and signaling pathways in innate immune cells, resulting in the production of pro-inflammatory cytokines during infection. However, Coxiella type 4 secretion activity suppresses many of these pathways, and previous work in the field have shown that Coxiella type 4 secretion system suppresses many pro-inflammatory cytokines such as TNF. So moving into this work, we wanted to test the activity of the type 4 secretion system in our hands and see if it suppresses other cytokines, as well as extend this work into primary cells. To do this, we differentiated THP1 monocytes or primary human monocytes into macrophages, and we infected these cells with a BSL-2 strain of wild-type Coxiella or Coxiella, type 4 secretion system Coxiella, that this mutant strain lacks the inability to suppress type 4 secretion system, type 4 secreted effectors into the whole cell. We then looked at mRNA expression of different pro-inflammatory cytokines to see whether there's a difference in pro-inflammatory cytokine expression. What I'm showing here today is that the THP1 mRNA data for IL-6, where, as you can see, when Coxiella loses its type 4 secretion system, we get an increase in IL-6 expression, demonstrating that this type 4 secretion activity is important for Coxiella to suppress IL-6 pro-inflammatory cytokines. And in data that I'm not showing you today, we see similar trends in other pro-inflammatory cytokines such as IL-1 beta and TNF. We also see similar trends extended into the primary human cells, where we see that the secretion of IL-6 is dependent on this type 4 secretion system together. Taken together, the type 4 secretion system of Coxiella is important for it to suppress these pro-inflammatory cytokines. Many of these pro-inflammatory cytokines converges upon NF-kappa B pathway, and NF-kappa B is an important transcription factor that induces many of these pro-inflammatory cytokines. Now, previous work that I've shown you before, there's 2 Coxiella effectors that suppress NF-kappa B signaling. However, Coxiella has a large repertoire of over 100 different effectors and a broad range of hosts that it infects. So we propose there's possible other effectors that may be blocking this NF-kappa B signaling pathway. To get at this, we utilize this NF-kappa B reporter screen where we express several type 4 secretion system infectors on mammalian expression vector. We then utilize this 5'-luciferase plasmid that is under the control of the NF-kappa B responsive elements promoter. We co-transfected these plasmid in HEC293 cells and then stimulated these cells with TNF and red luminescence as a readout for NF-kappa B activation. Of the panel of effectors that we saw, we identified a specific effector, CBU1314, that significantly suppresses NF-kappa B signaling compared to our control here in blue. This was very exciting to us because in data that I'm not showing you today, CBU13 suppresses NF-kappa B signaling downstream of multiple innate immune agonists such as IL-1 beta, and it does this in a dose-dependent manner. So we wanted to see and wanted to figure out what is this protein and if there's any literature in the Coxiella field that have worked or identified this protein. So we dived into the literature and what is CBU1314? CBU1314 localizes exclusively to the whole cell nucleus, as we could see right here in this microscopy image, where we have GFP expressed in CBU1314, and when we topically express these in cells, CBU1314 localizes exclusive to the whole cell nucleus. Additionally, from the literature, we know that CBU1314 tightly associates with chromatin and that it broadly modulates the whole transcriptome. Additionally, we know that CBU1314 associates with genes that are involved in host cell signaling, transcription, and host immune response. So taking what we know in the literature and what we've seen from our NF-kappa B reporter screen, we wanted to see what specific immune response is CBU1314 suppressing. To answer this question, we ectopically express CBU1314 in THP1 cells. And as a positive control, as I'm showing here in the data, we utilize mCherry as a positive control for gene expression, as well as utilize this negative control for gene expression, where we utilize this YOPJ effector. YOPJ is another protein effector, secreted effector from another pathogen, Yersinia tuberculosis, where it's a potent inhibitor for NF-kappa B signaling and pro-inflammatory gene expression. And as I'm showing you here, when we express CBU1314 in cells, we get a significant decrease in mRNA expression of pro-inflammatory cytokines, and I'm showing you TNF. But in data that I'm not showing you, I don't have to show it to you today, we see that CBU1314 significantly suppress other pro-inflammatory cytokines, such as IL-6. So taking this together, we wanted to figure out what host protein CBU1314 is potentially targeting in the host cell to suppress NF-kappa B signaling or immune gene expression, especially since it localizes exclusively to the host cell nucleus. And from the literature, many of these other effector proteins perturb a lot of these immune pathways through binding or interacting with specific host proteins. So to do this, we expressed CBU1314 or GFP in HEC293 cells and stimulated these cells with TNF. We then utilized IP mass spec to look for potential host protein targets. Looking at the mass spec data, two of the top hits from the mass spec data were these two host proteins, CD73 and PAF1. Surprisingly, these two proteins are a part of a larger complex known as the polymerase-associated factor 1 complex, and I'll be referring to it as PAF1 for the rest of this talk. So what is PAF1? Excitingly, we know and read through the literature, PAF1 is a highly conserved eukaryotic complex that is comprised of 6 subunits that all plays a part in how it functions. PAF1 plays a huge role in many transcriptional processes, such as histone modification, polyadenylation, and cleavage. But one of its well-studied functions is PAF1's role in transcriptional elongation, where it facilitates the recruitment or the proximal pause release of RNA polymerase II. So this was exciting to us because now we've identified this complex that is associated with transcription, and we know that CBU13 suppresses several immune genes. And we know from previous work with CBU1314 that it's associated with genes that are involved in transcription. So we wanted to understand the functions of PAF1 as it relates to immune response and Coxiella infection. And data that I don't have time to show you today, when we utilize our NF-kappa B reporter system, when we knock down PAF1 in cells, we see that PAF1 is crucial for NF-kappa B signaling. So additionally, to look at the functions of PAF1 as it relates to Coxiella infection, we knocked down PAF1 in mouse macrophages, and then we infected these mouse macrophages with wild-type Coxiella or Coxiella with our type 4 secretion system mutant Coxiella. We then look at mRNA expression of several pro-inflammatory cytokines to look at the functional relationship or the function of PAF1 as it relates to Coxiella infection. And what I'm showing you here is the mRNA expression data for TNF. And when we knocked down PAF1 in these mouse macrophages and we infected with wild type or the type 4 secretion system mutant, we see a significant decrease in the gene expression of TNF, suggesting that are telling us that PAF1 is contributing to Coxiella-induced immune gene expression in these primary cells. And then when we don't have this complex, we see a decrease in the expression of these genes. We see similar trends in other pro-inflammatory cytokines such as IL-1 and IL-1 beta and IL-1 alpha as well. Lastly, we wanted to understand and want to see whether PAF1 is important in restricting Coxiella in these same cell types. So to do this, we knocked down PAF1 in these mouse macrophages, and we infected these mouse macrophages with wild-type Coxiella. We then analyzed Coxiella growth over 7 days, and what we could see right here is that PAF1 significantly restricts Coxiella replication in these mouse macrophages, as we see a threefold increase in the replication when we knock down PAF1 compared to our siRNA control. So what I've shown you so far today is that Coxiella is a stealth pathogen that utilizes its type 4 secretion activity to secrete many different effectors into the whole cell. Many of these vectors play an important role in suppressing different immune pathways. However, this particular infector that localizes to the whole cell nucleus exclusively suppresses immune gene response and immune gene expression in the host immune responses, while it suppresses NF-kappa B signaling. Additionally, what I've shown you today is that CBU1314 interact with this transcription factor, PAF1, where it interacts with PAF1, and PAF1 is important for many different transcriptional processes, and PAF1 is also important to promote NF-kappa B signaling and promote gene expression. Additionally, I've shown you today is that this PAF1 complex is important for restricting Coxiella replication in primary cells. With that, we still have a lot of open questions about this interaction between PAF1 and CBU1314. And one of the key questions that I'm currently working on is what domain or residues are important for are required for CBU1314 to suppress NF-kappa B signaling, and also are these domains important for interaction with PAF1? To test this question is necessary as it will help develop tools and understand how CBU1314 may be affecting PAF1 function. And as mentioned before, I started analyzing and working on this, and I've begun to answer this question. And I've done this by generating truncated C-terminal domain and N-terminal domain mutants of CBU1314. I utilized my NF-kappa B reporter system to transfect these CBU1314 mutants in HEC293 cells and analyze luminescence as a readout of NF-kappa B signaling to analyze these different mutants to see whether we see a change in NF-kappa B suppression. In this graph I'm showing you right here, we have our M-cherry positive control for NF-kappa B activation. We also have our full-length CBU1314 protein and our YAP-J as a control for NF-kappa B suppression. In the first three mutants I'm showing you right here, we have C-terminal domain mutants where we mutated the last 4, 10, or 20 amino acids. And you can see right, and we also have 2 N-terminal domain mutants where we mutated the first 20 or the first 51 amino acids. I've also included here an NLS mutant where this CBU1314 mutant can no longer translocate it to the host cell nucleus. And as you can see right here, these residues 5 to 10, where we see an increase in NF-kappa B signaling, suggesting that these residues 5 to 10 are important to suppress for this protein to suppress NF-kappa B signaling. Additionally, we also see that CBU1314 is important for localization to the nucleus, is important for it to suppress NF-kappa B signaling. Additional questions that remain is whether CBU1314 is interacting with this PAF1 complex directly. And I'm currently utilizing a bacterial two hybrid approach to answer this question, in addition to potentially utilizing purified bacterial proteins, a PAF1 complex protein, to assess for direct interaction. Additional question is how is the CBU1314 manipulating PAF1 function? And we hypothesize here that CBU1314 may be antagonizing PAF1 function, and what function of PAF1 is antagonizing is left to be worked out. Is it blocking PAF1 recruitment of RNA polymerase II, or is it affecting other path-1 function as it relates to transcription? And one of the key direction that I know a lot of people is probably thinking is my talk after hearing my talk today is whether or not CBU 1314 is important for the growth of Coxiella in these cells, especially since Coxiella has other effectors that suppress NF-kappa B signaling. And with that said, I would like to thank my lab. I would like to thank my mentor, Dr. Sunny Shins, and I'll also like to thank Natasha Lopez-Fisher, who was the main driver for this project and generated a lot of the data that I've shown you today. Additionally, I'd like to thank my collaborators and all our funding sources in the lab. And to hear more about this story and updates on this story, you could always email me and I'll take questions.
Great job, Jadine. We have time for probably 1 or 2 questions if you want to leave them in the chat. All right, we have one here from Rachel. You might have mentioned this, but how does Coxiella evade LPS-dependent TLR2 or TLR4 signaling? Also, how does Coxiella evade lysosomal trafficking and autophagy? Are there autophagy-specific effectors?
Hi, Rachel. Yeah, so I'm not 100% sure of the specific mechanism, but as I mentioned before, Coxiella relies heavily on its type 4 secretion system effectors. So I wouldn't be surprised if Coxiella main way of evading many of these responses or TLR4, TLR2 responses is utilizing its type 4 secretion system effectors. What specific effectors, I'm not 100% sure of, but I'm guarantee you that it's probably utilizing many of these effectors to suppress that signaling pathway. And in regards to autophagy specific effectors, from previous work or like in the literature, Coxiella also have effectors that localizes to the host, to its Coxiella containing vacuole membrane. And a lot of these vacuolar localized membrane facilitates Coxiella ability to fuse with like lysosomes or autophagosomes. So many of these effectors are important for actually driving the CCV because it's a very fusogenic vacuole and it fuses with a lot of different like host membranes. So a lot of these effectors actually helps like the Coxiella containing vacuole like regulates its fusogenic property and allows it to regulate its pH and its ability to fuse with different host vesicles within the cell.
All right, I think that might be it for questions. Great job again.
Thank you.
Okay, and I will be introducing the last speaker for the day. So I have the honor of presenting our keynote speaker. He earned his PhD in microbiology at the University of Virginia and completed his postdoctoral work in Dr. Samuel Miller's lab at University of Washington. He is currently the professor and chair of the Department of Microbial Pathogenesis in University of Maryland, Baltimore. The focus of his lab is to understand the molecular basis by which bacteria, both gram-positive and negative, modify the lipid component of their membrane and how the modifications can affect or circumnavigate host immune response. His lab does so by utilizing different cutting-edge technologies, including but not limited to mass spectrometry, gas spectrometry, and bioinformatics. He has earned many honors and awards, most recently awarded as a Fellow of American Academy of Microbiology for 2024, Distinguished University Professor from University of Maryland in 2023, and Regents Awards for Excellence in Research from University of Maryland in 2021. Not to age him, but he has been a member of ASM since 1997 and served as the president of Maryland branch of ASM in 2022. He was also chair for Mid-Atlantic Microbial Pathogenesis meeting for 2022, which is a meeting that our lab and many of our branch members attend. Please join me in welcoming Dr. Robert Ernst.
Thank you very much for the very kind introduction. I will put a plug out for MAMPM. MAMPM will be happening in February. We have a limited number of people who can apply, 200 max. So once you see it, it would behoove you to get on the schedule. First, I'd like to thank the committee and the Eastern Branch for inviting me to give a talk today. I'm really sorry that I couldn't make it up there, but I do collaborate with a number of people up in that area, so I am sure I'll be up there someday in the future. Second of all, I don't mind being interrupted during my talk for questions, and that's perfectly fine. So if you have something that's pressing, feel free just to speak out. I can't see the chat on my screen right now, and it's full screen. So the title of the talk is Bacterial Lipids from Diagnostics to Immune Modulators. Let me see if I get it to. I have to do disclosure. Some of the work I'll talk to you about, we've started 2 small companies, one for bacterial diagnostics and one for adjuvant development, and we'll come to that date in a minute. But the overview here is to talk about bacterial lipids. I'll almost guarantee you that you know more about bacterial proteins, bacterial transcriptomics, and a lot of people just jump over the lipids, even though, especially in gram-negative bacteria, the outer membrane is basically composed of lipids. Then I'm going to talk about a microbial barcode. Can we use this lipid signature to tell you what the bacteria are? And in this case, I'll tell you how we do it and how we can do it directly from blood without culture anymore. Then I'm going to talk briefly about how we're looking at in vivo lipidae, because a lot of times when we grow things in a tube or on a plate, the lipidae will subtly change, and these subtle changes are actually very important for pathogenesis. And at the end, I'll talk how to make a TLR4 agonist, or basically a vaccine adjuvant. So when you think about proteins, it's really easy to understand structure. When you think about the RNA and DNA, you understand structure. But I'm going to show you that lipid structures are very variable. And a lot of times what you're going to have is you're going to have a series of head groups. In this case, it's choline or ethanolamine or glycerol. And then you're going to have various fatty acids down at the bottom. And phospholipids always have 2 fatty acids. And the standard, most normal fatty acids that you're going to find in bacteria are ones of 16 carbons in length, 18 carbons in length, or 16,1 and 18,1, meaning that they have an unsaturation back up in the middle of the fatty acid, usually between the 9th and the 10th carbon. We're not going to focus a lot on that. Gram-positive bacteria have something called cardiolipins. They also have LTA, lipid-teichoic acid, but the cardiolipins are much more prevalent. And what cardiolipins kind of look like is 2 phospholipids put together with a glycerol head group attaching. But what we're going to really spend a lot of time on is lipopolysaccharide. I am more of a gram-negative bacterial person than a gram-positive, but we're slowly moving to the gram-positive world now. So if we look at LPS, it's mainly in the outer leaflet of the outer membrane. Because of time, I cut out a couple slides. LPS, lipid biosynthesis, begins on the inner leaflet of the inner membrane, proceeds to the outer leaflet of the inner membrane, and then directly to the outer leaflet of the outer membrane through a very elegant system called the LPT operon. There's 3 major regions of LPS, from the cell here, which would be the blue part, all the way to outside the cell, the serotypes. So lipid A is also known as endotoxin, and it's the toxic portion of the cell. And the 2 blue dots are glucosamine, 6 carbon sugars. The white dots are phosphates, and the squiggly lines are various acyl chains, and we'll come back to that in a minute. Then what we're going to have is a series of 8 carbon sugars, 7 carbon sugars, and then everything else external as it goes out are 6 carbon sugars. I'll tell you now, for most of the stuff that we work on, this glycosidic linkage is very easy to cleave with a little acid and heat. I throw away the carbohydrates. If you're a carbohydrate person, you're amazing because the work out here is very difficult, and we're only going to focus on the lipids here. So when we do this analysis, E. coli has done us a service. There's literally approximately 10 to the 6 LPS molecules per cell. If the bacteria is replicating every 20 minutes, 70,000 new LPS molecules need to be transported to the surface every minute. So it's a very cost, carbohydrate intensive purpose. It constitutes between 75 and 95% of the entire outer leaflet of the outer membrane. Per bacterium, there's probably about 10 attimoles. So if you do a back of the hand calculation, 1 microgram of LPS is equivalent to all the LPS from 10 to the 9th bacteria being presented to the cell at once. So if you see studies, immunological studies, where people are using 1 microgram of LPS, most likely it does not mimic what you're going to see in a physiological infection. All right, that's just way too much because 10 to the 9th bacteria normally will kill an animal almost immediately. It serves as a permeability barrier, it's important in virulence, and it elicits a mammalian immune response, and we'll come back to that at the end. Over the years, when I first started doing this, everybody thought LPS was E. coli. E. coli shown here on the right side. 2-glucosamines, 2 phosphates, 6 fatty acids. On the backbone, all of these fatty acids are 14 carbons in length. 1 piggyback here on the 2' position is 14, and over here on the 3' is 12. This is the most pro-inflammatory LPS that I've ever seen and that we've ever identified. If you're looking at Coxiella or Pseudomonas or any other bacteria where you're trying to understand pro-inflammatory responses, you shouldn't be using E. coli because that's not the pro-inflammatory response your cells are seeing. You should be using LPS from the cells or the bacteria that you're studying. If this is the most pro-inflammatory molecule, in this case, we're only talking about human TLR4 because mouse and human TLR4 are slightly different. 2-glucosamines, 2 phosphates, 4 3-hydroxy-C14s, basically the same backbone. This molecule is 100% invisible to human TLR4 MD2. And then Pseudomonas, which has 5 fatty acids, it's a little more pro-inflammatory. And now H. pylori is again a tetra-acylated molecule with very long chain fatty acids. And this signals very poorly to not signaling in human cells. So there's variation in acyl chain, their position, their number. Some of these, then all these have terminal phosphates. So some bacteria will take off the terminal phosphates or put modifications on. And these are all influenced by disease state, temperature, cations, antimicrobial peptides, pH. And the more different bacteria you look at, the more chance you're going to find, especially if you get low passage clinical isolates from patients, you'll find something new. And we've seen this before, and a lot of people study it. Lipid A, and all you really need is lipid A. Coronoantigen is dispensable, at least in my opinion. Is TLR4 really the receptor for lipid A? Not really. It's really MD2. MD2 looks like basically a catcher's mitt. Lipid A fits down in. If it fits in perfectly, the phosphates align with positively charged residues on the surface of MD2. It changes its structure. TLR4 feels that change, and now it changes its structure, and then in theory, the tear domains change. The problem with my current working hypothesis is that all structural work on MD2 TLR4 is ectopic domain only. That's where the crystal structure came from in that Park paper. So we really don't know what happens to the tear domain depending on structure. And that's something we're trying to do now with folks who are doing cryo-EM. So we're going to switch back now. And in the beginning, we started to look at a lot of different structures. As it was pointed out, I've been a member of ASM for a very long time. When we started to get, it took me, don't smile, Shingo, I see that. When we wanted to get one spectra from beginning to end, took me 4 weeks and 1 to 2 liters of bacteria. Now we can do it from almost anything down to 1,000 bacteria in an hour. So we started to look at a lot of different bacteria, and we started to notice gram-negatives, gram-positive, and fungal pathogens all have unique lipid codes. just similar to what you see here as a barcode. So the perfect assay would be any pathogen, a quick isolation. This is just a very simple MALDI mass spec, and we're not going to have time to go in the theory of MALDI. It generates a spectra, refer it to a database, and it kicks out and it spits out, data. If you're a clinical microbiologist, Bruker Biotyper, bioMeri Uvitics, they do it on protein signature. Well, they have to have pure cultures. They can't do it directly from complex biological fluids. So how do we get over that? And what we did is we made this assay over the last 5 or 6 years. It's called FLAT. And basically, it's just a fast assay where we don't purify the lipids anymore. They go from whatever sample, in this case, it's shown as a plate, directly onto a metal MALDI plate. We apply a buffer. We cook it for 30 minutes at heat. We wash it. You put on matrix. You put it in a simple MALDI, and you're done. Beginning to end, hands-on time, 3 minutes. So now that we can do this really quickly, how do we do it safely? And one of the things we came up with this little device called the Panini Press, and it looks exactly what it sounds like. You put your samples in here. There's a little water in here. You close it. You put it in a standard heat block, and within 60 minutes, you have your data. So the nice thing about this is that we can work with BSL-2, BSL-3 pathogens. We can put this in a biosafety cabinet, and it works very easily. This is standard spectra from a gram-negative. In this case, it's Acinetobacter baumannii, a gram-positive Staph aureus, and fungi Candida albicans. We can clearly see that these spectra are completely different. And one of the cool things about it is, if you start mixing multiple bacteria together, we have no issues giving you polymicrobial infections. And this is a bigger deal when you're looking at clinical samples where you don't get to choose what comes in urine, what comes in blood, what comes in as a bronchoalveolar lavage fluid. And that's where we've been working now, is that I know we're not going to replace standard protein-based diagnostics. They're great. They work really well if you can grow them. But if you're looking at TB, which takes 21 days, that's not a very good assay. We can now do TB as soon as we can get a culture or a BAL or wherever it comes from in roughly an hour, hour and a half. Some of that's a little harder. This is fungal pathogens. Not only does it work for bacteria, but here's 3 different Candidas, so we can subspeciate. Here's our favorite Saccharomyces cerevisiae for a lot of good reasons. But then these are filamentous fungus. So one assay, all of these within an hour. So we've had many, many people in the lab working on this, and we're slowly branching this out. But one of the things that we've really done is that we've looked at escape pathogens, sepsis, UTI, emerging infections, Coxiella. I want Coxiella. I don't have that yet. So hopefully we can talk about getting cells. If you've got any weird and wonderful bacteria or pathogens in your lab, I'd love to talk. We're always looking for more. We are probably close to 600 or 700 different bacteria fungus, and we've only had a couple failures so far. So we're really looking for more. And then again, to really drive things going forward, where I want to be is direct without ex vivo growth. Because if you grow things in vitro, things change. So we've also tried to advance FLAT. We can now do MS to the MS. And now what used to take me months, Hyojik Yang in the lab, who is a mass spec expert, he does this in an hour and a half. From beginning to end, he can do structure. One colony, MS to the MS, and now we know, especially things like, where is a modification to these phosphates? Is it on the 4' on the left, or is it on 1' on the right? And these are important to try to understand how this affects antimicrobial resistance. And something that we're going to just start launching now is, how does it affect the entirety of the membrane? Because if you start to change structure or acylation, proteins change, lipids change, everything changes. And that's one of the things that hasn't been easy to do, but now we have the technology to do it. We're also trying to make it better, because we're always trying to do faster. So in this case, we can take 10 microliters of material, put it directly onto what is basically we call shark skin. And over the course of a couple of minutes, all of the sample will dry down into that little spot. So now we're down to a couple thousand bacteria instead of 10 to the 4. So now what we can do is, we know that things like UTIs are considered at 10 to the 5th. We know we can do that right now easily, but can we do 10 to the fourth and 10 to the 3 to be able to give the clinician a response on what their patient may have so that they can treat them quicker with, with better antibiotic stewardship. And this is where we launched into. I'll just show you some data that's pretty recent on bloodstream infections. So there's lots of cases of bloodstream infections, and basically 1 in 8 people die from sepsis. The number of cases are increasing year by year. And what happens is that there's really only a small number of bacteria that are normally present. There's probably 10, 15 that we really have to isolate. And the gram-positive and the gram-negative ones here are shown, E. coli, clab, Pseudomonas, staph, and faecalis. So this is what the normal scheme is to get down to protein-based biotyping. Sample goes into a blood bottle. It grows from 18 to 36 hours in the blood bottle before it turns positive. It has to be plated directly, and it has to grow again before they can go ahead and do their assay. So we want to skip this, go directly to here. And this is the entirety of the assay. Blood bottle's positive, get rid of the red cells for 30 seconds, pellet all the rest of the material. And it doesn't really matter if there's cells carried forward, eukaryotic cells. We put it on the multiplate and we do it again. So now this takes 5 minutes, this takes 60 minutes. And this is the spectra that we see directly from blood. Signal to noise is excellent. We see here's staph, and this staph looks a little more complicated. And these are the cardiolipins and the spectra I showed before. Here's E. coli. Here's E. coli with modifications. In this case, we know what every one of these ions represents structurally. And then here's E. faecalis, which is a different gram net positive. So what I didn't show you here is that we have all the fungal and we have a lot of other bacteria that we can do right now very simply. This is way too many bacteria. This is 10 to the 8th bacteria, 10 to the 9th bacteria. So these blood cultures grew 12 to 15 hours. The goal now is, can we grow blood cultures for 4 hours and do the same exact analysis, thereby, again, taking off days for the ability of clinicians to treat their patients more judiciously. So this is just a synopsis of here. And where we're working with now, we can do the same exact thing with urine. Actually, urine's a lot easier than blood. We're down to 10 to the 5th, 10 to the 4th. We're working on BL now, which literally go in and do bronchovelar lavage or sputum, do the sampling, do the processing directly. And currently, if the samples are really hot, meaning they have a lot of bacteria, we can do it now, but we need to reduce the limit of detection on that. Now, the question that we ask a lot is, what happens in vivo? And before a lot of our techniques, we couldn't do anything in vivo because we just didn't have a whole lot of bacteria. So one of the bacteria dear to my heart is Pseudomonas aeruginosa, and I've been working on this for a long time. And it's found everywhere, soil, water, and it causes a lot of infections. But for what we're going to talk about today, it really causes airway infections in people with cystic fibrosis. And greater than 60% of adults with CF have Pseudomonas eventually. It used to come earlier. Now it's being identified later, which is a sign that CF clinicians and nutritionists and all these other people are making CF patients better. So we did a very simple experiment. We asked if we take PAK and PaO1 are just laboratory adapted isolates, if we infect animals with quite a few bacteria, we know that now, we collect their lungs or their BAL 8 hours later, so this is acute infection, and we did lipid analysis and extraction analysis directly. When you look at what went in vivo, so what you're looking at here are just very basic multispectra. This is mass on the lower axis. On the vertical axis is relative intensity. MALDI is not quantitative. So big peaks are big peaks and little peaks are little peaks. So let's not try to overinterpret on this, because I'll show you data that is more quantitative. So what went in is a molecule here, and I'll show you structure later, this is a penta-acylated molecule with 1 hydroxyl group, an OH group, and 2. If you look at lug homogenates or BALs, this is just ground up, and this is just a true lavage. We're now seeing that we see a second set of ions that are bigger, and these represent hexa-acylated molecules. Again, 1 hydroxyl group and 2, and the reason why I'm saying this is important, I'll come back later. So what happens is, in culture, it makes this molecule where there's 5 fatty acids on it, and this position, this 3 position, is deacylated by an enzyme called PAGL. And all these mass differences are really easy to determine what the structure is, but we also can do MS of the MS. What happens in vivo, that PAGL, the enzyme, we've done transcriptomics. This is Casey Hofstadter's work. He just graduated recently, is back in the clinics finishing up his MD. Transcriptionally, it's turned off in vivo. And then the enzymes responsible for putting on these hydroxylases, one of them is turned on, increased in vivo, and one of them is neutral, because that's the one that's involved in here. So no matter what we did, whether it was lung homogeneous or BAL, we get this same pattern where things are changing. Why is this important? This is exactly what we're seeing in the lungs of this is exactly what we're seeing with organisms that are collected from the airways of people with cystic fibrosis. They are making these exact modifications. And how do we know this? Well, we recapitulated this in a non-destructive assay in a mouse model. So one of the things that my lab has been doing, along with a true mass spec imagery person, Alison Scott, in our department, is we're, instead of being destructive, we can leave the architecture in place, take that piece of tissue, so take out the airways, the lung heart of the animal. What we do is we fill this with gelatin, we fill the trachea, and we inflate the lungs, take it out, we freeze it, so now the airways are still open, cryosection, 13 micrometer piece of tissue, coat it with matrix, put it in this case, it's called the timsTOF, a Bruker timsTOF, and then over the surface of that piece of tissue, we raster a laser. And at some distance, whether it's 200 microns, 50 microns, 10 microns, we collect a spectra, we summate those into a single spectra, and we tell the computer, put back where you found that ion, and let's see where it went to. So what I'm showing you now is just a histological this is an H&E stain, one lung here, heart here. You can see that the airways are open. We stained these airways with Ly6G to show where the neutrophils are. So the neutrophils are down in the lower part of this of these animals' airways. Why are they there and up instead of up in the parenchyma? Because the intranasal installation makes larger particles, and what happens is it does sit down where the bronchi come out at the bottom. And there's other methods for aerosolizing, but we're still developing this assay for future use. So when I showed you here, this is all the data we got when we did it by the destructive method. So what you're seeing here on the right here is this area, on the left here is this area. These are the same exact 2 ions as I showed you on the previous MALDI. This is penta-acylated with 1 hydroxyl group. This is penta-acylated with 2 hydroxyl group. This is exactly where we're seeing this increase in staining for neutrophils. And this is known to be one of the hallmarks of Pseudomonas infections, is neutrophils are where the Pseudomonas are, but Pseudomonas is pretty resistant to neutrophil killing for a lot of reasons that we won't have time to talk about today. So this is the penta-acylated. Now I'm going to switch. This is the hexa. So the same thing here, 1 hydroxyl group, 2 hydroxyl groups. Look how, I'm going to go back and forth real quickly. This is much farther out into the lung than you're seeing with the penta. So what you're seeing here is that the deacylation mainly occurs deeper into the infection for reasons that we don't know yet. And again, this is really nice. This is at 50 microns, so the laser is hitting every 50 microns. And the level of resolution doesn't really allow us to do a lot of in-depth analysis. This is now down at 10 microns, the laser's hitting. This clearly shows deacylation is only occurring deep in the infection, deep in the biofilm, where PAGL is not active for reasons unknown still out in the periphery of the infection. So what we're trying to do for this now is we can go in and try to do laser capture. We can try to go in and see what's actually there. But one of the cool things about mass spec imaging is I'm not just collecting data on these 4 ions. I'm collecting data from, in this case, 1,000 mass units to 2,400 mass units. We'll be able to see cytokines. We'll be able to see other lipids. We'll be able to see other bacterial products potentially, because really all we're doing is looking at the lipids now because of the matrix, but you can do lipids, metabolites, cytokines, proteins, you have to convert them first into peptides. And all of this is now doable with this technique. A lot of mass spec imaging has been used for looking at tissues, looking at other things, but there are very few people who actually take it and do pathogenesis. And that's really Alison Scott's specialty here.
Hey, Bob, do you mind if I ask a question?
Absolutely.
How long does this 10 micron resolution take for like this small section of lung?
A couple hours.
Okay.
Well, so Allison just put on, it's called a microgrid onto the instrument. We can go down in theory to 1 to 2 microns. So you can scan this much at low, this much at medium, and this much that's tiny.
Mm.
But you can take serial sections. And as long as you take serial sections, you can use one and you can do the next thing. Next thing is, like, after I do mass spec imaging, I can strip off the matrix and I can counterstain it, with an H&E or something like that. So we can marry them together. The other thing that's happened that's really cool is that there's companies now making antibodies or FAB tagged fragments to certain parts of whatever immune cells with a metal tag. You target it on your cell, you expose the tissue, the labeled tissue with, in this case, it's kind of UV light. The metal falls off, and you don't image where the actual immune cell is, you image where the metal went. And you can put 10 or 20 different antibodies on a single piece of tissue and get them all at the same time. So there's a lot that can be done here. How do we know that what we're looking at is actually what I'm saying? Again, this is another Hyojik Yang experiment where he can now do MS to the MS on tissue, which is incredibly interesting because now I don't have to worry about what's happening ex vivo. I'm absolutely positive that what I'm looking at in vivo is what we say it is. We still have issues with quantitation in this method too. But what you can do is if you have a known standard, especially have a standard that matches to whatever metabolite, lipid or whatever, you can spot it on the tissue and you can do the analysis there. And then your ion intensity can be matched up by doing a linear regression-ish. This is the instrument, and I will say that one of the things that were either brilliant or wildly stupid is we put it into a BSL-3. So now we don't have to kill SARS-CoV infected lungs. We don't have to kill TB infected tissue. And that's the only setup in the world right now that we know of. So what it does, what I'm really trying to do is, I'm a select agent lab 2 here, and I'd like to convert this lab instead of just BSL-3 to a select agent BSL-3 lab because there's so many experiments that I want to do with Yersinia. So now I'm going to switch over to how do we make a TLR4 agonist. This is about what half my lab does. Because we studied structure, we knew that various structures resulted in that were non-stimulatory or highly stimulatory. So we came up with a method which allowed us to make lipid A molecules that never existed before. I think I'm going to skip, yeah. And so this is something we published a long time ago called bacterial enzymatic combinatorial chemistry. Literally, it enables us to make lipid A molecules as quickly as 10 to 14 days, where we can clone any lipid A biosynthetic enzyme from any organisms. And we've got this from 10 or 15 different organisms now. We put them into any bacteria that we want. We culture, we extract the lipid A, look at the lipid A structure, and we can do biochemical, well, this was 10 to 14 days. Now from step A to E now takes about 4 days. And if we find that we have the structure of this novel lipid A, we then can check whether it's in vitro and in vivo. So the nice thing about this is that we screened hundreds of new molecules through THP cells, through HEK cells, THP cells, primary splenocytes from mice, primary PBMCs from humans, human DCs. And we tried to find molecules that had altered innate immune responses with the idea, can they be used as a vaccine adjuvant? And I'll explain why that in a second. So we spent years doing this, and we got really, really good, and we screened. But the problem being is if you want to make a product, one of the things you have to do is biological molecules are very hard to make reproducibly, and they're very difficult to be approved through FDA mechanisms. So over the last 4 or 5 years, through money that we've received from NIH through contracting mechanisms, we've had the ability to make synthetic molecules, purely synthetic molecules, which gets rid of congener distribution and makes a more straightforward IND path for potential use in a vaccine someday in the future. These are the two molecules that we came up with. They're called BEC 438S and BEC 470S. The S designation for synthetic, 2 glucosamines, 6 fatty acids, and either 1 or 2 phosphates. This gets us back to here. The bacteria that we started with, in my infinitely smart approach, was avirulent Yersinia pestis, because why not start out with a molecule that makes the most minimal lipid A, and we can start modifying accordingly. One of the things that I'm not going to be able to talk about today is that one of the earliest evolutionary processes from Yersinia pseudotuberculosis to Yersinia pestis was the loss of an enzyme called PAGL, which puts on a long chain fatty acid here, a C16. It's located in the outer membrane, and for reasons we still haven't been able to figure out, why did Yersinia pestis break this enzyme? One of it is that if this enzyme is in place, it makes the lipid A molecule recognizable to the human host MD2-TLR4. So one of the first things that I did was to fix this. And lo and behold, it put a C16 fatty acid back on here. And then if we grow Yersinia cold, it puts on an unsaturated fatty acid here. Well, the only difference that I'm going to show you again, as I said, is that we've also we found an enzyme, or an enzyme was identified to specifically remove this 4' position fatty acid phosphate. And if you look, I'm going to jump forward. We know it's going to stimulate through TLR4. Here's E. coli in black. Here's FAD, and I'll show you the structure of FAD, which is another synthetic molecule. And here's our 2 different structures. So these are THP1 blue cells. These are stimulated with vitamin D3 to get them to stick onto the tissue culture plate instead of PMA. And then this is just quanti blue. So we always do our stimulations over a very large stimulation range, from 100 nanograms down to 100 nanograms down to 10 picograms. And what you can see is that each one of these different molecules had a different pro-inflammatory potential. And this is exactly what we were looking for, something that had an EC50 driven over to the right by a log, 2 logs, even more, because we didn't want these to be too pro-inflammatory, because obviously this is endotoxin. Why did we think this was a viable idea? Because GSK is the current owner of MPL, monophosphorylated lipid A, which was purchased by Corixa, which was made by Rebe. And this is a biologically derived molecule, and this is in thousands and thousands and thousands of different vaccines, Shingrix, Fendrix. A couple of these things are different vaccines on the market that have been used. So what you're seeing here is 2 phosphates, two glucosamines, a phosphate over here. This side, the 1' fatty acid is moved, and there's this configuration of between 3 and 7 fatty acids. So it's a mixture, and it's a wild mixture, because this is the congener distribution that's allowed by the FDA. Plus or minus 35% is acceptable for this molecule. Crota, which has also been known as Avanti, Crota brought Avanti during the pandemic. This is a synthetic molecule where now all the fatty acids are 14 carbons in length, and the same fatty acid and the same phosphate group is removed as FAD. But what you can see, all of these subtle differences result in different pro-inflammatory responses. So one of the things that we did is we made these molecules and we gave them out to all the friends that I possibly knew that would want to play with them. We've done a lot of bacterial antigens. We've done a lot of viral antigens. And because of time, I'm not going to be able to go through all of these. But what we can do, what I can say is, for reasons really unknown, how about, right, 470 seems to be more efficacious with protein for our viral antigens and bacterial toxins, whereas 438 seems to be more efficacious with bacterial antigens. Why is this? We don't know yet, because we don't know what these subtle differences do. But we know that it's the C16-1 when it fits down into the MD2 pocket, has this little kink in it. And I think that subtle change changes the changes the way that MD2 changes its structure. And that's the reason why these two BECs seem to be excellent potential candidates for future use in a vaccine someday. We also formulate. And that's one of the things that's really big now. If I take just, well, I'm not going to do it because I'll make you guys spastic. If you take just the BEC alone, that is not allowable as a vaccine formulation. We call it admix, and we use it a lot. But in all vaccines, you have to mix it with some other component that can then be used. So one of the ideas that we're using is, what else can we do to make our vaccines better? Shockingly, these molecules have no toxicity, even though they're endotoxins in rabbits, because that would be an issue. And one of the things, I'm not going to show you how we got to this, one of the things that's different from our adjuvant, the BECs, versus all the others, is that we get a balanced Th1, Th2 response in all of the different models that we're looking at. So we get IgG1 and IgG2 A and C, depending on which animal background we're using. We also get IgG2B and IgG3, which we don't really understand at this point. It works via intramuscular route of infection or vaccination, intranasal, and this is really new from a couple of weeks ago, microneedles, and I'll show you data on that. So far, it works in mice, rats, dogs, rabbits. It went into calves a couple of weeks ago. We get antigen sparing and dose sparing, and antigen sparing by 1,000-fold. So for some of the things that we're doing, say in flu, we're down to nanograms amount of protein instead of microgram. It gives you T-cell responses, memory, and then again, if you want to make an interesting vaccine, it's very stable. So two of the models I'm going to show you right now, because this is some of our latest data that I don't think anyone's seen yet. There's avian flu, high-path avian flu, and then there's standard run-of-the-mill influenza, which is H1. We've published a lot on H1, and I'm going to show you more on data on H5. Do the BECs, in this case 470, work to adjuvant H5 to protect animals? In this case, it's a mouse model. We do use, so we're using HA from H5. We're using it at very slow doses, and our comparator is FAD versus the BECs. We do things a little differently when we do our vaccinations. So our prime boosts are not 21 days apart. They're only 14, and we've gone down as short as a week for certain models with getting FSK with the BECs. So not only are we looking at immune responses, but we're also looking at challenge. So all of our models always have a challenge model. In this case, to mimic an H5 infection, because you need a high-path lab to do that, a BSL-3 lab, which we don't have, you can use a PR8 backbone pseudotype with a Vietnam 04 in an intranasal infection. Because of time, I'm not going to go through all the data. We have weight loss data, we have isotyping data, but the most simple data that's the easiest to see. Here's the controls, where it's just the protein alone, we see significant death. Here's when we put in FAD at 2 different concentrations of the H5. They can say at high doses, FAD works very well. At low doses, FAD loses more than 60% of the animals. And the BECs, on the other hand, show no death, show no weight loss, and show, again, a balanced Th1, Th2 response. Why is this a little surprising? Because the normal human vaccines right now that are licensed for H5 use 90 micrograms of antigen, because the H5 HA is not very immunogenic. So this is all brand new. This is work by Devin Ellison, a PhD in the lab, who's shared between myself and Matt Freeman's group. And then this is some more really, really interesting data that we're coming out now. This is work with a group down at College Park, Chris Jewell's group. In this case, dissolvable microneedles. A patch that you push on your skin, no needles, no blood, no pain. Can we use our molecules with HA alone and see what happens? In this case, we don't have a challenge model because these are very early data. Again, we're back to HA, a very small amount of it. And these are the microneedles. So these are made by Camilla Edwards and Smitty Oaks and Chris Jewell's lab. So it's 127 needle configuration. You can see a microneedle over here. These are the actual physical microneedles. And what you can see here is that the needle itself is here, and it's about It's about 1 millimeter, roughly. It's dissolvable. So in this tip is the Beck and the antigen backfilled with gelatin. And then the top is filled with more gelatin to make the structure that you see here. So literally all you have to do is it goes onto the flank of the animal. The fur is clipped with a standard clipper. It sits on the skin for up to 60 minutes and they're self-adhering. Basically, they go on a Velcro mechanism. And if they don't fall off by themselves, we go and pull them off. So in this case, it was up to 60 minutes, but we know we have data where we misunderstood what we were supposed to do. We put it on and took it off in 10 seconds. And we get very similar results, which is kind of cool. So what you're seeing here, this is total IgG, and these are HA-specific titers. On the left, here's the microneedles, and on the right is day 28. So this is the prime and the boost. The red is the microneedles at high dose. The green is the microneedles at low dose, and the purple here is the control of intramuscular immunization. What you're seeing here for total IgG is prime alone gives you almost maximal responses that you don't get at lower doses, and you clearly don't get intramuscular. And your animal-to-animal variability is all but gone compared to the rest of it. What's really cool here's IgG1 at day 14 and 28, IgG2 at day 14 and 28, and you get maximal responses by intramuscular immunization by the prime alone. One of the expertises of Chris Jewell's group is they can watch things now go to the draining lymph node. Because we can do mass spec imaging, I can find our Beck molecules, if they're in sufficient quantity, from the skin to the draining lymph node. And we can then, if we can see them in the draining lymph node, we can now look to see what might be going on around them. We did this back in the day with Francisella in the spleen. And we can clearly see at the interface between the red pulp where Francisella is and the white pulp where the immune response was going on, we saw arachidonic acid coming off one specific phospholipid and that being converted into signaling lipids. So there's a lot of things that we can do in this case. This takes an army. I have really, really, really wonderful people. Sayen is doing the microneedles and the vaccinology. Ian is moving into that area. Moira, Sydney, Brandon, Linda are exquisite technicians who are trained in just about all of it. These are the folks, a lot of these were people who have just graduated. You can see one person here who you may recognize his name. That is now in Sonny and Igor's group. We have a lot of friends. We also have a lot of fun. This is the Guinness Brewery outside of Baltimore. I thank Baltimore, NIH, Cystic Fibrosis Foundation for all their funding. And like everybody else, we are looking for postdocs and have a lot of opportunities in a lot of different areas. So thank you very much, and I'll take questions. I know it's a lot and I speak fast. I apologize. I try not to speak fast, but it's not within me.
Okay, thank you for the presentation. We have a couple questions in the chat.
Okay.
I've made Kishore able to speak, so if you'd like to ask your question now.
Yes, 1 second, I'll put my video on. Dr. Ernst, I wanted to hear your talk for the last 5-6 years, but finally, EPA made it. But I really wanted to see you in person.
Mhm.
It's a brilliant work that you're doing. Actually, Dieter Schieferle, one of the ASM EPA member, was actually telling me that, Kishore, you should talk to Bob. So it's an amazing presentation you gave on the diagnosis. I think it's very, very convincing. On the adjuvant part, that was very, very dear and interesting to me because I started an adjuvant company, just left my academic position after 20 years. So I will really catch up with you.
Okay.
But the key point I wanted to ask you is that you talked about a lot about the lipid modifications, but do bacteria alter the 4' and 1' phosphate groups?
Yep, lots of them do. Francisella, where we stole those enzymes from, make LPXE and MLPXF. Porphyromonas gingivalis, one of the causative agents of cavities. When they go in as a pro-inflammatory lipid A, as they recognize heme activates a couple of these phosphatases. to then make themselves go invisible because they've already caused damage. They've already set up, their niche. Now they don't want to be, eradicated or found by the immune system. There's a lot of bacteria that are modifying the terminal phosphates, because if the terminal phosphates are negatively charged, you can put a phosphethanolamine or aminorhabinose, and now you're changing the overall negative charge on the surface of cells. And I didn't talk about that today. We have lots and lots of data showing that that's important for things like polymyxin resistance or any cationic antimicrobial peptide. So
that's very interesting. So I wanted to ask about another phosphate related question. It's, the MPLA, the readjuvant, and what is GSK approved, or what is the, the CRODA and everything, is one phosphate is out. It's MPLA. But you showed amazing data with the DPLA, with both phosphates. One of the one that you actually putting on the left-hand side, I think, at least from. That was very, very interesting. So what is your rationale or explanation for going for the DPLA? Because when you go for the FDA approval and all those kind of things, because the MPLA is kind of becoming the mainstream, right? Like you talked about the Pendrix and even Cervarix, and so which one, if I were to ask you a very stupid, simple kid question, which one would you pick between the two, the MPLA or the DPLA version?
Oh, mine. Nobody can get GSK's version of MPL. You can get Invivagen's, and it's made in two different ways, and Invivagen doesn't tell you that. So there are subtle differences. We normally have MPL built into a lot of our assays, but we don't do it anymore because we don't use the biologic. And our head-to-heads, we exceed everything from MPL, or Vivagen's MPL. Why can we see the diphosphorylated? We don't know. That's a really weird thing that we found. And as I said, it's trending between different antigens. If you put it on LAL, Endosafe, it has endotoxic activity like MPL does and all the other ones. But I will tell you, which makes no sense to me. Well, it makes sense to me because I probably have a good idea why. The 470 has almost no LAL activity.
Wow.
Well, LAL is a horrible assay. It doesn't measure anything that we know. So we don't know if it recognizes an LPS that's pro-inflammatory or completely nothing. And I think we finally broke it. Someday, if I see you in person, I'll tell you a conversation I had with Jack Levine, the guy who made LAL.
Thank you, Dr. Ernst. Okay.
Bob's fine, please.
Okay, and then we have one other question on the chat. So Vincent asked, so thank you for the wonderful talk. In the context of therapeutic efficacy or interaction in polymicrobial infections, can you use internal standards with the flat assay to quantify bacterial loads?
Yes, and that's where we're moving now. What we're doing is I have some synthetic molecules that I have, or I can incorporate other synthetic molecules or controls. And what you do is you start to dope them into your sample. But what you do is you start out with a standard amount of your internal standard and increasing amounts of bacteria. And if you do that over and over with different bacteria, you draw a line with your internal standard, and you can do, I know semi-quantitative is a silly term, but that's what we use a lot. We'll get a semi-quantitative assay right now. In the case of UTI infections or urine, we're better than, because of our limited detection is right where they need it to say whether it's a UTI or not. We can, with better than 99% accuracy, I can tell you a negative. If we do it and then it goes off to culture, we'll be able to tell you what's a negative, and then in an hour I'll be able to tell you what the positive is too. And that's, I think, one of the weird things, that because the assay is right at the place that we need it for UTIs, which is right between 10 to the 4, 10 to the 5th. That's right. Sometimes it's better just even if I can't, even if I don't identify the positives, reducing the workload for a clinical technician or a clinical lab to throw away the negatives so that they don't have to work them up is also valuable, because I don't know if there's any clinical technicians here, or people who run clinical labs. People who work in the labs there are overworked all the time because samples don't stop coming.
Okay, great. So 2 other questions right now. So this one is from Dr. Bitaro. So back to the diagnosis. In relation to gram-positives, do you see subspecies lipid patterns? For example, species with commensal and pathogen members.
Sometimes yes and sometimes no. But the problem being is I don't have a lot of gram-positives. So when I work up the gram-negatives, when I did all the escape pathogens, I have a collaborator, Yohei Doi, at the University of Pittsburgh. I get 700 Acinetobacter, 700 Klebs. Someone's got 700 staphs or streps, let me know. I'll take them all. Because we can plow through those in 2 to 3 weeks, especially if they're on plates. You know, that's the beauty now, is that we're now we've got the easy ones down. Can we go ahead and find some of the weirder ones? You know, we're looking at things coming from the Chesapeake Bay in collaboration with Charlie Sweet from the Naval Academy, of bacteria that grow at 10 degrees below the thermocline. And the things we're seeing there are absolutely bonkers cool.
Okay, and then the one last question that I see is from Ling Wu. In regards to the multi-TOF work with clinical samples, have you correlated your data, organism identification, with bacterial growth from clinical blood cultures?
Yep.
They are curious if multi-TOF might identify organisms that do not end up growing in blood cultures itself.
That's going to be a hard one, because 1 to 10 organisms per mil in a patient is septic and probably lethal. We're not PCR. We really can't we can't go down to the amplification of PCR. So we can do anaerobes, we can do any of the blood bottles are fine, whether the peds or they're adults, whether there's various resins in, it doesn't seem to matter, but we still need some replication. So that's where we're really excited about BAL, because that's going to come right out of the patient, or sputums, or UTI, or urine. So we do see polymicrobials. We'll see a lot of commensal. We'll see like staph epidermidis or things like that, but then we'll see a bloom of, you know, Serratia or Proteus or something else that's a lot easier to see.
All right, so I think that is all for our questions today. Thank you all for attending this 811th meeting of EPAASM, and thank you for the flexibility with this last minute change in format. Thank you to all our fantastic speakers as well for sharing your excellent research with us. And one last announcement before we let everybody go for the night. This meeting is the last major event with Shingo and I as co-presidents and members of the EPAASM board. As we start events over the summer and the next academic year, Zach Walter and Dan Netting, who you heard speak earlier from Jefferson, are taking over as co-presidents, with Adam Glass from Drexel stepping up as vice president, Jadine and Amber from Penn remaining on the board as treasurer and secretary, and then Shingo and I will be replaced by a new board member, Anita Cowell from Vincent's Lab, who we'd like to thank for stepping up to represent Temple in the student board. Thank you all so much for your support this past year, and we want to wish the best of luck to the new board and to all the fantastic events they have planned for the next year. So thank you all for coming, and have a great rest of your night.
Thank you.